and we will let people roll on in here. If you're just getting in, drop us a line in the chat where you're tuning in from tonight. We always like to hear where folks are attending from. And we'll give it a couple minutes before we begin. Let people come in and connect. For our attendees, the format of this talk, we won't be able to hear you, but we can read your chat and Q&A boxes. So I would encourage you to tune in, let us know in the chat box where you're coming in from, and drop us questions throughout the evening so that I can field those for our panelists. And we'll just wait a few more minutes here to let people come on in. Great, well, why don't we get started with the introductions? Welcome to Intersections of Environment and Justice, From Our Bodies to the Earth. This webinar series is brought to you by Biosphere 2 and Arizona Institutes for Resilience. My name is Aaron Bouget, and I'll be the moderator this evening. I'm moderating from Biosphere 2. You can see some of the historic space frame behind me. And it's a pleasure to be here and has been for the last 3 weeks or so. This webinar series brings together the fall 2020 Carson Scholars cohort of graduate students to discuss the future of food, management and reuse of resources, environmental, health and social justice, and environmental stresses and climate impacts. Our 12 scholars over the last 3 weeks detail the challenges facing communities around the world and innovations that could impact the way we interact with the planet and each other. There we go. Today's webinar is titled De-Stressing the Earth and Ourselves, and we have 3 panelists joining us this evening. Alex Karnish is a third-year PhD student in Ecology and Evolutionary Biology. He studies mutualisms, species interactions that benefit both species involve and how resources are allocated to them. He does this by measuring leaf, flower, and fruit traits of the local Sonoran plants, including a native chili pepper, the chiltepine. JoRee LaFrance comes from the Absaluga Crow Nation and was born and raised on the Crow Reservation in southeastern Montana. She is a third-year PhD student at the University of Arizona in the Department of Environmental Sciences. JoRee's research aims to understand water quality issues in the Little Bighorn River watershed and those impacts on the indigenous food, energy, water, security, and sovereignty nexus and tribal water uses on her reservation. And lastly, Grace Windler is a longtime resident of the Southwest, having grown up in New Mexico and moved here to Tucson for graduate school. As a PhD student in the field of paleoclimatology, Grace uses fossil molecules found in marine sediments to reconstruct past changes in temperature and rainfall. Particularly, she aims to understand how the tropics have responded in the past to large shifts in Earth's climate state. Welcome, Alex, JoRee, and Grace. And lastly, before we begin, I'd like to thank our Carson Scholars Program donors and our University of Arizona sponsors, Biosphere 2, College of Engineering, College of Science, College of Behavioral Sciences, and the Graduate College. And last but not least, I'd also like to recognize our webinar mentors, Kevin Bonine, Chris Kokinos, Andrea Gerlach, and special thank you to Maya Patterson, who's been working magic in the administrative behind the scenes role. Thank you, Maya. And lastly, before we get started, I just want to remind all of our attendees that we're going to have our panelists present back to back to back, during which we encourage you to ask questions in the Q&A box, and I'll be fielding those questions to our panelists at the start of our discussion. So we'll do 3 presentations in a row, and then we'll move into the discussion period. So drop us a question, and with that said, I'll pass it over to Alex.
Thank you, Aaron. Let me get my screen up. Hi, everyone. As Aaron said, I'm Alex Karnish, and today I'll be talking about how plants allocate their resources to different traits by using a local pepper that you may be familiar with. So in our lives, we all have priorities that we need to send time and energy to keep them up. So some of us, or all of us, need to put time and energy into our jobs to get to work and make money. We need to put time and energy into our family and friends to maintain those good relationships. And if we have some energy left over, we'll put that into hobbies to make us happy. So when times are good and you have plenty of time or energy, you're able to do all of these 3 pretty well. But when times get stressful and you don't have as much time on your hands or you get really tired, you might need to let one of these go so you can keep the others functioning. So I know when finals gets really hard for me, I usually need to drop off my hobbies so I can maintain my work and keep up my relationships with my family and friends. Similarly, plants have priorities. So instead of time and energy, they have nutrients and water. And so they split this nutrients and water similarly amongst 3 priorities, just like us. Instead of work, they put resources into growth, so making stems, roots, and leaves. Instead of making family and friends, they put resources into reproduction, so making flowers and seeds. And instead of hobbies, plants can put resources into defense. So that can be chemical defenses in their tissue, or as this example is right here with these cactus spines, physical defenses. So I'm interested in what is the most important priority? What is the order of these? For plants, is growth the highest priority and then reproduction, then defense, or is it some other combination of those? And so I do that by looking at 3 local species that you might be familiar with. I look at 2 species of Jimson weed or moonflower, so Datura rhytii and Datura discolor. And then I also use the local chili pepper, the chiltepin, which I will be presenting on today. And so this chili is also known as Capsicum annuum, and it grows right around here in southern Arizona. And so a little more in depth on these chiltepins, they're the ancestor of a lot of the peppers that we eat. They are found just south of Tucson, Arizona. They produce these nice little white flowers with 6 petals. They're pollinated by bees that need to come up, and the buzzing of the bees is what pollinates them. And then once those get pollinated, they produce these bright red pea-sized fruits. And so as we all know, chili fruits are spicy. And so that is the main chemical that I'm looking at for how chilies allocate resources to their defense. And so just to talk about capsaicin, once more, the chemical that makes these fruits spicy. So the way we measure capsaicin is through the Scoville heat scale using Scoville heat units. So on the far left here, we have 0 SHU with our friend the bell pepper. It's not spicy at all. It doesn't have any capsaicin. And then on the far right, we have military grade pepper spray, which can have over 3 million Scoville heat units. So it's a really high concentration of capsaicin. So to give you an idea of where the chiltepin lands on this scale, I have a jalapeno pepper here as reference. So your average jalapeno has about 5,000 SHU. It's moderately spicy. And so the chiltepin is roughly 20 times hotter than a jalapeno. So even though this fruit is pretty much the size of a pea, it's pretty small, it's very, very spicy. So the way I study this is by growing plants in situations where they have very few resources and where they have plenty of resources. And then I see which are reduced in size or magnitude in order to figure out which have high priority and which have low priority. And so to do this, I'm looking at their defense, reproduction, and growth. So I measure their growth by looking at the size and number of their leaves. I measure their reproduction by looking at the size and number of flowers they produce, as well as the amount of nectar that they put in those flowers to reward the bees. And then to measure their defenses, I see how spicy their fruits are. So usually I do this by sending fruits to a lab in New Mexico, but sometimes I can do it quickly just by eating them. And so you're probably wondering what the consequences of these resource allocation rules could possibly be. And so these plants don't just interact with themselves, they're part of a broader network of species interactions. So you can see here, this bee is drinking the nectar from the flowers and pollinating them. This bird is eating the fruits and then moving those seeds around and depositing them to grow the next generation of plants. There's a ton of insect interactions that are going on the surface of the plant. You might have ants that are defending the plant from herbivores. You might have caterpillars or leaf suckers that are eating away at the leaves and wood. And there are an innumerable amount of below ground interactions between fungi and bacteria and other microbes that can both help and hinder the plants. And so you can imagine a situation where maybe the environment shifts. And since flowers aren't a high priority and there are fewer resources, the plants produce fewer flowers. So that means there's less food for those bees. So if the bees are really food limited, you might have a local extinction event for those bees that interact with this plant. And the same goes for this bird. So the birds that eat the fruit, and chiltepins are bird dispersed. Birds eat the fruits and then move the seeds around. Um, if fruit aren't as appetizing to birds, or if they just don't produce as many fruit, that can impact the next generation of plants that grow. And so overall, I just want to leave you with some take-home points about this. So plants have priority rules for how to spend their resources, just like we do. And how they spend those resources does change their leaves, flowers, and fruits. And then how they change those leaves, flowers, and fruits in response to nutrient or resource limitation can have broad effects on the species that interact with them. And with that, I'd like to thank you for listening to my presentation. I'm looking forward to all of your questions that you have. And I'll hand it over to JoRee, who's going to take this from kind of this individual species or plant scale and scale it up to a whole watershed.
Shora, Balaja Eugene Machlushuk Ichikdalum. Hello everyone, my name is JoRee LaFrance. My Apsalagan name is Eugene Machlish, which means fortunate with horses. I come from the Apsalagan Nation in Montana, but I currently reside here in Tucson, Arizona, which is on the lands of the Tohono O'odham and Pascoyaqui Nations. I'm a third-year PhD student in the Department of Environmental Sciences. Today, I will be speaking to you about Bemachbe, sacred waters, and how I'm bridging traditional understandings and hydrologic science to protect the Little Bighorn River. But first and foremost, before I begin speaking about my research, I'd just like to thank and acknowledge all those who have poured their knowledge into me and who have supported me along the way. For that, I'm super and forever grateful. A little bit of background. The Crow Reservation is located in southeastern Montana. It's about 2.3 million acres in size and is home to Apsalagat people who are also known as the children of the large-beaked bird, which is the people that I come from. We have about 14,000 enrolled tribal members, but about 70% of them live on the reservation relying on these three main watersheds, the Bighorn River, the Little Bighorn River, and Arrow Creek watersheds. Apsalagat people were known for our exquisite beadwork, using geometric and floral designs, and our ability to use our colors in those designs. We're also known for the teepee capital of the world, which happens on the banks of the Little Bighorn River. We're also known for the Bighorn River, which is a world-class fishing river, and we're very proud to come from the land that we come from. The Little Bighorn River watershed is located on the eastern end of the reservation. On the top left photo, we can see the watershed delineated in green. The headwaters of the Little Bighorn River begins in Wyoming in the Bighorn Mountains, and it flows north about 142 miles, and it converges into the Bighorn River, which is at the northeastern end of our reservation. The river is used by both tribal and non-tribal users. I can only speak for my people, and I know that we use this river for many, many different purposes, ceremonial, cultural, recreational, domestic, municipal. We have many reservations. Actually, most of our reservation towns are located on the Little Bighorn River, and Crow Agency, which is our main town, actually diverts water from the river itself and treats it in its plant and then disperses it into the community. So that just gives you an idea of how much this river is used. I grew up along this river, just a walk not too far from my house, and I had this pretty naive assumption that this river was pure and pristine. But it wasn't until my little sister became ill after swimming in the river that I really started to question what was actually in these waters. And with the Crow Environmental Health Steering Committee, I was able to become more aware that we had these various contamination hotspots, and we can see those in these red stars on the map on the right. These are things like feedlot discharge points, wastewater discharge points. We have abandoned mines in this vicinity in this watershed, and then we have agriculturally developed lands all along the riparian corridors of this river. So my people, we've lived along this river for many generations, and as Apsalaga, we derive from the water and land itself. We're a product of the land and our connection to it. We're taught that in our creation story, First Maker molded us from mud from the deep, deep waters. And during that time when the land was being created, Red Woman carved the streams, the lakes, the rivers for the water to flow. And then we started to learn, living along this land, that we were instructed by the 7 sacred rams to never change the names of these rivers, the Bighorn rivers, Little Bighorn rivers, that as long as we keep these names, that we will always remain as Apsalagat people. And these are just some of those teachings that we know about our responsibility to these rivers. And to me, the river is a reflection of who we are. And when we're not healthy, when our river is not healthy, we're not healthy. And I've taken the responsibility of taking care of this river a much, much deeper level. So taking those teachings and many values and those traditional uses of the river, with my research, I have to attribute much of it to the motivation and support from my family and my people, because if it had not been for them, I would not be where I am. And so beginning with that support and coming to the University of Arizona and beginning a collaboration with the Crow Environmental Health Steering Committee, I developed a sampling plan in the Little Bighorn River and was able to obtain funding, super grateful for that. And I began the collection process after creating this plan and being able to get it approved by our steering committee and our research center on our reservation. I collected samples from the Little Bighorn River, and then after that collection, I come back to the University of Arizona and process those samples for them to be read in the instruments. After that, we have to do more analysis with that data to actually get the concentration numbers. And then with that data, we can eventually go into disseminating our results and getting a bigger picture of what's happening in the river. Like I mentioned before, my study site is the entire Little Bighorn River. I have about 22 sample locations beginning near the headwaters and ending towards the convergence of the Little Bighorn River to the Bighorn River. These sample locations were picked strategically based on tribal access location and their proximity to these contamination hotspots that I mentioned earlier. And specifically on the hydrology end of my research, I am trying to understand how pollutants are behaving at these different times of the year. So I actually went to go sample in December, and then I went and sampled this past June. And so I had 2 different times of the year data where I was able to compare and see how these contaminants may be varying or behaving as the water changes, because we know that moisture is a reflection of that season and how much we received that season. And so December, we're getting base flow, and June, because of snow melt, that river, or the water is ending up in the river. So we're starting to increase our flow, and I want to understand how these pollutants are behaving in that. So I do all of this by first collecting samples in the river. The joys of doing my research at home is that I'm able to share this experience with my family and actually collect samples on our own lands. Fun fact, one of my samples is located in Medicine Tail Coulee, which is actually directly where the Battle of Little Bighorn happened. And so this just goes to show you how close to home all of this is for me. And at each location, I do collect 4 water samples and then use a device to collect various parameters that I use later on. Each day, because I'm collecting across the whole river, I'm only about to collect 7 samples a day. So I actually have to ship my samples every single day back to the University of Arizona. So when I'm all finished in Montana, I come back to Arizona and come back to the Arizona Laboratory for Emerging Contaminants, where I'm able to start the process of preparing my organic samples to be read in instruments. Thank you and huge shout out to Alec for their employees for running my inorganic samples. And so as you can see that it's a long process to actually be able to get these larger samples down to these smaller vials that we're able to put into the instrument to be read. And then we have to do more calibration and work on a computer to get the actual concentration. So ultimately, with my work, I know that this is much bigger than me, and I want to rely on our traditional understandings to guide my research and what I do. I'm still in this learning process, but ultimately, I just hope that my research will contribute to the efforts of my tribe obtaining treatment and state status. And what that means is a tribe is able to set their own water quality standards on the basis of cultural uses. And that's important because we just heard this talk about how my people use the river in different ways that might not be recognized or used in ways that other people use water. And so it's very important that we know these cultural uses, but also find data and be able to back that data up. So me as an Upsalogat tribal member, I've taken that responsibility on, and the goal for my research is to just contribute to the efforts or ignite that effort to obtain that treatment of state status so we're able to continue to use these rivers just as our ancestors did. I believe that our kids deserve the same quality of water and deserve to be able to use these same waters just as our people have done for many, many generations. Ohogajila, thank you very much for listening. Thank you to my advisors, my funders, and all those who have supported me along the way. I appreciate you all. And now that we've heard a little bit about the impacts to the environment at a watershed scale, we'll get to hear a little bit more from Grace, who will take us up another notch and looking at the global scale. Thank you so much.
Okay, thanks, JoRee. All right, so as Erin mentioned, my name is Grace Windler, and I'm at in the Department of Geosciences here at U of A. And today I want to talk a little bit about how I use fossil molecules in my research to understand past changes in Earth's climate. And before I sort of get at what fossil molecules are, which some of you may be wondering, first I want to take a step back from Alex and JoRee's presentations and show you this global picture of average sea surface temperatures. And it kind of looks like how you might expect. So the water up at the high latitudes in the North and South poles are cooler, and the water gets warmer near the equator. But what I really want to draw your attention to here is this sort of bullseye dark red centered around this area between Southeast Asia and Australia. So this region is known as the Indo-Pacific warm pool, and it has the warmest sea surface temperatures on Earth, really. And you know, waters in this region can get up to about 90 degrees Fahrenheit. And just for reference, that's within the recommended range to keep your hot tub. So this water can get pretty warm, right? And since it's so warm, it evaporates really easily and rises into the atmosphere. So this region is sort of acting like a giant jacuzzi. At least that's sometimes how I like to think of it. And if we look at this from a different perspective, so here I'm showing you sort of a cartoon schematic of a cross-section of the atmosphere along the equator. And just to orient us, here is Australia right here. You can see that above that warm pool area, there's just this really large column of water vapor rising up into the atmosphere. And once it's up there, it gets distributed to other parts of the planet. And so this area is actually sort of the single largest source of heat and moisture into the global atmosphere. And it's not hard to imagine that changes in this circulation could have a global impact. And one sort of modern example of this that some of us might be familiar with is a little thing called El Nino. So El Nino events sort of start by changes in this circulation over the West Pacific. And so this is a really important area for understanding changes in Earth's climate. And in my own research, what I focused on is using fossil molecules to understand potentially how this circulation has changed in the past under sort of large shifts in Earth's climate. So shifts between sort of interglacial warm periods like the one that we're in today and glacial cool periods during which sort of there were these massive ice sheets sitting over the northern hemisphere. And in this figure here, I'm showing you the difference of what this region looks like geographically between these two climate states. And so on the left, this is what this area looks like today. You can see there's a lot of islands there. Whereas on the right, this is what this area looked like at the last glacial maximum, so the most recent glacial period. And what happens is during glacial periods, a lot of water is taken up by ice that's sitting over the northern hemisphere. And so sea level is a lot lower. And you can see that this low sea level actually exposes a lot of this area between these islands. And now it's continent during these periods. And so this big change in the geography, we think actually contributed to changes in that circulation that we see over the worm pool today. And so this is where molecules come into play, right? So these are molecules made by ancient life, which can actually tell us about ancient climate that the organisms that made these molecules were living in, right? And so these are molecules like fats, waxes, or oils that are made by organisms from, single-celled organisms similar to bacteria, all the way up to more complex things like plants. And today I really want to focus on one in particular. And it's a molecule made by this little guy. So this is a plankton. It's called a coccolithophore. And this plankton lives on the surface of the ocean. So it uses sunlight to photosynthesize its food. And we know that this plankton makes a very specific kind of molecule called an alkanone, right? Don't get too bogged down with the structure. It might give you flashbacks to chemistry classes, that's fine. But these molecules, these alkanones, they are sort of long chains of hydrogens bonded with carbons, right? So these are really strong bonds. And so the molecules themselves actually outlast the plankton. And what scientists have found is that the structure of these molecules made by this plankton actually varies with the temperature that the plankton lives in. So if we measure the ratio of the structure of these molecules in our samples, we can actually calculate a temperature. And so, now you might be wondering, how do we access these fossil molecules? And one of the ways we can do that is by drilling the ocean for sediment, right? So I use marine sediment cores, and the way those are collected is by these sort of large international teams of scientists who all get together on these large research vessels like the one pictured here. And they cruise around and use this big rig that's here in the middle to basically push a giant tube into the bottom of the ocean at pre-planned locations and pull up sediment. And what they're pulling up with that sediment is all of the stuff that's buried in there, including molecules. And so what this looks like is, at the end are these sort of long tubes of layered mud where the top of the core is younger sediment and the bottom of the core is older sediment that's been buried for a longer period of time. And so if we sample the sediment from these cores at different depths, we can get an idea of how things have changed through time, right? And unfortunately, I have not had the opportunity to actually go on one of these cruises and collect these cores, but rather once they're collected, they go into essentially a large library of cores, a repository. And, I can choose one I'd like samples from, pick the depths I want, and then I basically get a large box of mud, bags of mud in the mail, which is nice for me. It's always a nice day when I get some samples. And so now, we have our sediment samples, but you might be wondering, how do we go from what's essentially a bunch of mud to our molecules that we're interested in? And the way we do that is with this instrument that's pictured here on the left in our lab. So this is what's called an accelerated solvent extractor, or an ACE for short. And it basically works like a giant espresso machine. So instead of coffee grounds, each one of these little metal canisters up here is filled with a sediment sample. So it has ground up sediment in it. And one by one, the machine takes it in, and instead of water, it pumps through organic solvent through that sediment. So these are liquid compounds that filter through the sediment, and they dissolve all of the organic molecules that are in there. And then they collect down here in these large vials at the bottom. And what we get are sort of these solutions that are just kind of like a molecule soup, right? It just has all the molecules from each sediment sample in there. And we separate out the different types of molecules that we're interested in using this technique called column chromatography that I unfortunately don't have time to go into, but it's, as you can imagine from this picture, a bit of a hassle. And once they're separated, we can then put our samples on our machines and measure the molecules that we're most interested in. And just to remind everybody, so I've kind of done this process using a sediment core from right here where their star is off the coast of southern Sumatra, within that warm pool area, to reconstruct sea surface temperatures going backward in time. And at the end of this process, we get something that looks like this, a really long squiggly line. Sometimes I feel like my life is spent just staring at squiggles, right? And so just to help you all out here, so this is sea surface temperatures from that core I mentioned going backward in time. So you can think of this 0 on the left as the present or near present day, and it goes back to about 450,000 years into the past. And what you might notice from this is that there are periods in the past when the water was about as warm as it is today, but there are also several periods when it's several degrees cooler. And these are actually glacial periods, and they happen about every 100,000 years. But it's really neat that we can actually see this tropical surface water is showing these glacial shifts, right? And so this record that I've shown you here is just a really small, piece of a larger body of work from many different research groups that have, point at the fact that we think that during these cooler glacial periods, the circulation over the warm pool seems to have weakened over the Indian Ocean side of the warm pool. Whereas there's evidence to suggest that it actually got stronger over the Pacific Ocean side. So this like single column view that we think of today over this area, it sort of split and it became decoupled, right? And so, just with this very short example, I hope that I've kind of illustrated for you all that fossil molecules are a really useful tool for providing clues about how Earth's climate has changed in the past, and reconstructing past climate changes are really useful for understanding overall how our climate system works and responds to stresses. So with that, I'll say thank you for listening, and I will throw it back over to Aaron for the Q&A.
Thank you all for your great presentations. I would encourage our attendees now to ask some questions in the Q&A box. That'll be in your attendee panel. And we've got a question for JoRee from Alexandra to start us off. JoRee, thank you for your presentation. Was there opposition when wanting to conduct water sampling on your reservation? If so, what did you do?
Well, I didn't have opposition to do any sampling, but I guess I would say that water sampling isn't necessarily a priority for my tribe specifically, but I'm so thankful that we have groups like the Crow Environmental Health Steering Committee, Little Bighorn College, the Upsalogat Water and Wastewater Authority. We still have people who are concerned for our waters, whether that be surface water or groundwater. So in that sense, I didn't get any opposition, but it wasn't a huge priority for my tribe.
And JoRee, I have a follow-up question for you as well. You mentioned that your tribe might have relationships to the river that might be different from, other cultures, other societies. Could you lean into that a little bit, if you're comfortable, to describe what some of those relationships or activities might be?
Yeah, so one of the examples that I think most of my people do is just that showing that basic respect of feeding the river. And so this was instructed and taught to us, before my mom can even remember. So these are just teachings that have been passed down to us. So just showing that level of respect and feeding the river for Buluksat and to ask for protection, but also this is one area I would like to possibly pursue with some other tribal researchers from back home, is to look at the amount of sweat lodges that are along the Little Bighorn River. And so my people, we use the sweat lodge, and most of our sweat lodges at some of my locations as well, I've seen those lodges set up just right next to the river, and we actually take a bucket from the river and heat that water up and use that water, and then we go and bathe in the river, and then we drink water too. And so we use the river for other ceremonial purposes, which I'm not going to get into, but those are just some of the ways that we have a different connection that I've seen to this river.
Thank you. We have a question from Schuler. Excuse me, I'm sorry if I butchered that name. Thank you for your amazing presentations. All of your work has important implications for a wide variety of audiences. After the Carson Scholars program concludes, how do you anticipate sharing your research findings with the public during the pandemic and beyond? And Alex, do you want to start with that one?
Yeah, I'll jump right in. So I'm really grateful to be in Tucson. A lot of people are just really interested in the plants that grow around here. There isn't like any gathering I go to where I say I work with Datura or chiltepins that people don't immediately get interested in it. So I'm anticipating once the pandemic kind of slows down, there's a great local plant society that I can't wait to present at, because botanists are always a great crowd. And then I'm eventually hoping to maybe developing some relationships with the native seed search in Tucson, where I got a bunch of my chiltepin seeds, because they're just interested in what we do with their plants. Beyond that, I try to be active on Twitter. Other than that, I will think about that more.
Grace, do you want to chime in here?
Yeah, sure. So that's a really good question. So I think that the pandemic will maybe outlast my own PhD journey potentially. So I'm trying to finish in the spring, but I do think that there's a lot more opportunities for virtual outreach, such as this webinar, that I could potentially look for. And, I would continue to sort of look for opportunities in what are sort of referred to as science cafes. Sometimes local coffee shops or breweries tend to hold presentations for scientists and grad students in particular to do things like this. Yeah, and I think that, my research beyond my PhD might sort of focus a little more on more recent climate. So, uh, it's kind of up in the air right now, but I'll definitely continue to look for opportunities wherever they come up. I think that, yeah, the pandemic might outlast me a little bit in regards to my graduate career here.
And JoRee, do you want to chime in? What would communicating with your tribe and people outside of the tribe look like during these times?
Yeah, certainly. I think one of the main priorities for me, especially in my research, is ensuring that I'm able to communicate it to my people. So using much of the resources that Carson has provided, as well as some other fellowships, I've actually started working on a video to follow along my sampling and my lab work journey, and then just to give a little bit more information about the river itself. So just, I really want to find creative ways to disseminate my research, because I think that if I'm the only one who's able to understand this and my people can't, then what is the point of all of this? And so I take science communication very seriously, and I'm continuing to work to be better at it.
Thank you. We have a question from Diana. A rather political question, she says. Informed by your scientific research, what would you want the Biden or next presidential administration to do as a priority for the environment and science in its first 100 days?
I will start with that. The biggest suggestion, recommendation, I'll yell it, listen to Indigenous people. There's a lot of Indigenous experts out there, whether you're looking for knowledge holders from these communities themselves or experts in academia. Listen to Indigenous peoples, especially when it comes to climate change and anything science. Listen to Indigenous people is what I have to say. Thank you.
Grace or Alex?
Yeah, I'll jump in. I guess I just will say, taking climate change seriously is very important, and rejoining the Paris Climate Agreement is very important to, at least in my view personally. And, taking these large steps towards policies that encourage shifting our energy sources over as well. Anything in that realm would be great, hypothetically.
Alex?
I'll echo both of them. Climate change is a big priority, and saying that it is a real thing that is happening. And echoing JoRee, as listening to Indigenous peoples that lived here for thousands of years, they know how to manage the land. Another big thing for me is maintaining our natural areas and strengthening protections for them and not allowing industry into them. They have a lot of biodiversity, and they're actually very important for keeping our world clean and safe.
Thank you. We have a question from John Trorover for Grace. Thanks for a very interesting presentation. How are the seawater temperatures you measure with the alkanones translated into the global scale patterns of ocean-derived evaporation? Does your team collaborate with other teams to enable that upscaling of your molecular data to the global atmosphere? You might want to unpack that a little bit, Grace.
Yeah, there was a lot in there, but I'll say that once we have sort of these temperature reconstructions, and the example I gave is the Alkanone SST reconstructions, sea surface temperatures, there are a lot of different ways in which people look at data, paleoclimate data in particular. And, one single record from one place as your question gets at, is not going to really tell you very much. And so there are sort of these large scale community efforts to look at large networks of records in conjunction with one another, and look at sort of, depending on the time period you're interested in, like the last glacial maximum, for instance, which I mentioned, looking at what, you know, a lot of these records are saying from different parts of the world is one way to get at this. And another really important direction that I think the paleoclimate research community is headed is also looking at these large networks of reconstruction data in conjunction with model data, and like looking at what climate models are sort of physically based, telling you is happening in the climate based on, and that's based on physics. So you can look at whether the models agree with the data, where they disagree, and sort of see, you know, draw from that to understand sort of what might be happening physically in the climate system. I hope that gets at your question.
Thanks, Grace. Alex, I have a question for you. Kind of in the context of Grace's work, when you think about long-term changes in the climate and the environment, changes that might affect our resource allocation of plants, right? The choices that you're kind of looking into, that these plants are making. Have you thought about your research in the context of longer term climate change? And can you talk about maybe assumptions that you might make, or projections that you might make with the relationships to some of this behavior that you're researching?
Yeah, certainly. So I generally kind of just approach my work from a more conceptual point of view, not necessarily looking at or anticipating what could happen in the future. But there is some work, at least with the two species of Datura that I use, the Jimson weed, that water availability is actually very important for flower production and even like flower size. So these have large flowers. They can be 15 to 20 centimeters long. They're about this big, tube-shaped, and they can be significantly smaller when they don't receive as much water. And so for at least some of these plants in the Southwest, water seems to be a much more limiting resource, and a resource that affects resource allocation more than the others do.
Thank you. We have a question from Neil Brock. The Southwest is expected to be much warmer in the near future. Does the fossil-based historical thermal measurements agree with this? I think this is geared towards you, Grace, due to the changes in atmospheric mixing cells. And I can repeat that as well if you'd like.
Well, you know, I'm not entirely sure what is meant by atmospheric mixing cells, unless you're talking about sort of grid cells in a climate model. But a lot of work in the Western US and the Southwest in tree rings has been done, I know, to reconstruct sort of past wet and dry periods and droughts of the past. And we do know that sort of in the last couple thousand years, at least, there have been some droughts that have been pretty strong. But we know we're headed in a place that's going to go beyond that, I think. And in these tree ring records, you know, some of them, it depends on which records you're looking at, but some are water sensitive to precipitation, and some tell you more about temperature. And so, you know, it's a little outside my area of expertise, but I will say that there it's been a lot of work done reconstructing climate from the Western United States for the last couple thousand years. Yeah.
Grace, I have a follow-up to this. In a recent podcast on the Biosphere 2 podcast, I interviewed Dr. Brian Black, who's in the tree ring lab. And he actually pieces together data from tree rings, but also clams, dead clam shells from the past in what is called sclerochronology, as opposed to dendrochronology, which is tree rings. And my question for you is, have you considered kind of cross-referencing and cross-dating some of the trends you're seeing in your sediment samples with other similar reconstructions in the sclero and dendro communities? I know that piecing these different samples together is kind of an ongoing community practice.
Yeah, so that gets at a really important point, and something that is kind of a constant struggle for some people like myself. So the thing you need to think about when you compare different types of reconstructions, depending on the material you use, is sort of the time resolution that's represented by these different types of reconstructions. And so, you know, each data point in my sea surface temperature record, for instance, is I have a data point that's roughly occurs every, you know, 2,500 years is sort of my resolution, going back 450,000 years in the past. Whereas these sort of tree rings or the bivalves that you mentioned, the clams, when you look at the layers in the clam shells, these can be yearly resolutions. So these are much shorter timescales. And so it's really hard to kind of compare the longer timescales that I work on with something that's really finely resolved like that. Yeah, so it is difficult, but you know, there are other types of reconstructions that I can kind of look at what they say that go back to, you know, the last glacial maximum or beyond in sort of my general region to kind of look at that kind of thing. But it is something you need to think about when you are comparing reconstructions from the past, is like what sort of time frames you're looking at and can they really be compared? Yeah, hopefully that answered the question.
Yeah, thank you. Yeah. We have a question from Kevin. I appreciated the way you three transitioned across scales between presentations. How can researchers best communicate to the public and or younger students in the implications of scale? JoRee, do you want to start with this one?
Sure. So I guess the first thing that comes to my mind is my own research and thinking about the indigenous food, energy, water security, and sovereignty nexus, and thinking about the interconnectedness of everything. And so although things are at different scales, so you can think about our research, for example, the temperature global scales that Grace is studying is going to impact the water at watershed scales, and then this impacts of one single river, and then we can go about plants that are right next to the river. And so there's these interconnectedness, although these scales might be different, we need to think about these connections between these different things and know that they have roles and responsibilities, and each one is important, no matter how big or no matter how small, and knowing that they have implications for one another.
And Alex, do you want to chime in as somebody who maybe studies more at the individual conceptual level?
Yeah, so there was a survey a couple of years ago of ecologists in academia about like what ecological terms are the most important or the ecological concepts they considered the most important. And the top one was scale. So I think it's really important to kind of emphasize, or at least when learning about the importance of scale, to emphasize the importance of individual scale mechanisms and how those kind of all build on top of each other to eventually describe something much larger. I tend to think of it in like terms of little models for like, okay, this is how we would model a single species population over time. And then once you go from that, you go, okay, we have a community of species. How do they interact now? And it's all based on how that individual responds to the environment in the moment. So I think it's important to kind of emphasize that when you talk about scale, that you go from the smallest kind of to the biggest, and think about the things that you might lose as you go up in scale, but how they're kind of made by all these smaller pieces.
Yeah, I don't know if there's too much that I can really add to either of their answers there. but yeah, I would agree that it's important to sort of start at a small scale and then kind of apply these principles scaled up, especially, you know, sometimes I think on a more global scale. And when you think of like the global atmosphere, for instance, which is not, you know, necessarily what I specialize in, but when you think of the global atmosphere, for instance, it's interconnected, right? And so when you think of something happening in one place, it could affect something else happening in another place. So it is important to kind of start small and then go big, I think.
And we've got a question for JoRee from Raven. Beautiful presentation, she says. What has been the most surprising or alarming contaminant that you have found present in the Little Bighorn River?
Yeah, well, so far I've just been able to really dive into my inorganic data. So that's more like your toxic metals, your heavy metals, like arsenic and uranium. And so we do know that arsenic and uranium is present in the river. They're not at super alarming levels when we're thinking about it in terms of drinking water and EPA standards, but it is present, and that's still alarming for me. But I guess it just depends on where you stand with that. Although I have seen a little bit of my organic data, we're still working through that. And we did see some levels of pesticides in the river, and that is kind of a deal breaker for me, because I know if we know that it's in there, we know that contamination is happening, and we can start moving forward in developing an argument to move forward with these standards or the efforts of setting these water quality standards. So I'm really interested in my organic data, although really thankful to have my inorganic data to inform what's happening in terms of like mineralogy, geology, but really looking forward to seeing what's happening with my organic data.
And maybe the final question, a follow-up for you, JoRee. I have to say that the photos that you shared of you on horseback were stunning. And my question is, have you thought about doing sampling along the river via horseback, or would that be doing too much?
I've thought about it. No, it would be a little too much. Although, like I did mention, at one of my locations, my horses were in that pasture. So, but yeah, I tried all of the pictures in my presentation. I tried to put at the river, and so the beginning picture is actually my twin sister and I on our horses with our altitude dresses and our regalia by the river. And so it's not impossible, but I don't know if I'll be doing it for sampling.
Well, thank you. Yeah, it's absolutely beautiful. And thanks for sharing those photos with us. And
Thanks so
thank
much.
you to all three of our panelists for sharing their research, answering our questions, and presenting with us. And on a more general note, this does mark the last of our 4-part series. So I want to thank all of our attendees who have been with us all month. Thank you again for showing up and being here and listening to these great Carson Scholars. So tune in to future Carson Scholar programming, and thanks again, everybody, for a great night and a great webinar series. Good night, Grace. Good night, JoRee. Good night, Alex.